Hi folks, I thought I would try and capture what we went over during the study session today. So if you are not able to make it, I am hopefully going to be able to produce this video for you to kind of explain some of the practice problems that we went over. So I have added the practice problems under Unit 4. So if you found this video, you probably found the practice problems as well. So you can go through them and then watch this video afterwards to kind of check your answers. So this first one, let's look at the first question. It says, if tall plants are dominant to short plants. So the very first thing that I want to do here is to set my letters. So the question is telling you which is dominant and which is recessive. So it's saying that tall is dominant. So I'm going to use a capital T for my dominant allele and a lowercase t to represent my recessive allele, which will be for short plants. All right, so tall plants are dominant to short plants. So big T for tall, little t for short. It says a homozygous tall plant is crossed with a homozygous recessive plant. So homozygous tall is our genotype. So if it's homozygous, it has two alleles that are the same. And since it's tall, it will have two capital T's. And we're crossing this with a recessive, a homozygous recessive plant. So once again, we have two alleles that are the same, but it's the recessive allele, so we'll have two lowercase t's. So that's our two parents in this case. Now it asks what percent of the offspring will have the recessive allele. So now what we want to do is we want to place our parents across the top of our Punnett square and down the side. So I'll place one capital T in that block and our other capital T in that block from our first parent, and then I'll use the recessive alleles along the side. The four squares in the middle of our Punnett square represent our offspring. So basically we're saying if we bring down this parent's gamete, let's say this is the egg cell that's going to be fertilized by the sperm cell, then the offspring that are produced will have a combination of both parents. Same thing, we've got this gamete that's going to be fertilized by this one, so we're going to bring it across and bring it down, and we'll have that genotype. Same thing for the bottom blocks, we're going to bring that allele straight down and bring this allele over. And then our last block, we'll bring the big T down and bring the lowercase t across. So in this case, it asks what percent of the offspring will be homozygous recessive. We do not have any homozygous recessive offspring. So our answer really is 0, right? 0%. Now, if the question was asking you what percent of the offspring are heterozygous, then we would have 100%, because all of these are heterozygous, okay? If we are asking what percent of the offspring have a recessive allele, this one has a recessive, this one has a recessive, this one has a recessive, and this one has a recessive. So then it would also be 100% in that case. All right, so let's move on to question 2. It says if color blindness is an X-linked recessive condition. So here we have color blindness being an X-linked. So the previous question was not X-linked at all. We didn't mention anything about X-link. All right, so only apply the X and Y chromosomes to a question where it says X-linked is part of the condition. All right, don't make the assumption that it's there. So we have colorblind. So I'm going to use the letter A because I don't like C's. C's can be hard to tell the difference between a capital and a lowercase. So it says it's a recessive condition. So if they have the capital letter, they will not be colorblind. And if we got the lowercase letter, they will be colorblind. Okay. So we've set out our letters, but we know it's X-linked. So we have a man that is colorblind. So we're going to use an X and a Y to note our male. And we know that he is colorblind, and this is X-linked. So we need to place something on the X chromosome. And we're going to place the lowercase a since it represents colorblind. We're not going to place anything on the Y chromosome because it is an X-linked disorder. The Y chromosome does not have a corresponding allele on it. It's a different type of chromosome. And then the question says, a colorblind man has children with a woman who is a carrier for colorblindness. So the woman is going to have 2 X chromosomes, and she is a carrier for colorblindness. So to be a carrier, she has to have the recessive allele, but she doesn't show it. So we have a capital letter A on her other X chromosome. So she's not colorblind, but she can pass the recessive allele on to the next generation. So that is her genotype. So we're asking the question, will any of their daughters be colorblind? All right, so what I'm going to do here is kind of push this down onto the next page so we can see it all together. All right, so now I'm simply going to write my parents across one side. The X and the A's go together there, and I'll place my Y all by itself in that square. X capital A for this parent, and then an X lowercase a. So once again, I'll take my alleles and bring them down and bring them across to fill in the Punnett square. Oops. X capital A Y. All right, so what we see here is out of the daughters, because our question is asking, will any of their daughters be colorblind? So yes, we do have a colorblind daughter right here. Since it's recessive controlled, she has to have 2 recessive alleles or be homozygous recessive for colorblindness. So out of the daughters, 50% of the daughters will be colorblind in this case. Now, if we look over here for the male, there's also a colorblind male, right? So it just depends upon the question that we're asking. So if we look at the phenotype of this individual, phenotype, remember, means the physical expression of the genotype. So in this case, this individual is not colorblind. They have the capital letter A. This individual is not colorblind. They have the capital letter. This individual is colorblind, and this one is colorblind as well. Okay. So our question was, will any of the daughters be colorblind? Yes, 50% of the daughters will be colorblind. Now question B asks, can a male be a carrier for colorblindness? So remember, for males, they have the X and the Y chromosome. So can he be a carrier? Well, if you put the recessive allele beside the X chromosome, then this individual will be colorblind. Well, what if we put the capital letter beside our X? Now that individual's not colorblind, which is one of the criteria for being the carrier, but he does not have another allele on the Y chromosome to be able to pass it on. So a male cannot be a carrier for colorblindness because they only have one X chromosome. It says in question C, are males or females more likely to be affected by the X chromosome? Well, if we go back to B here, you can see that males are definitely more likely to be affected by an X-linked disorder. So males either have the disorder or don't have the disorder, whereas a female has many more genotypes that she could have, right? She could be all these different genotypes. So she could not have it, she could not have it, or she could have it, right? So 2 out of 3 gives her the normal color vision, whereas in males, it's 1/2, right? So there's 2 different genotypes that a female can have versus males. they only have one possibility, all right? So females can be carriers, males can be not, and therefore males are more likely to be affected by an X-linked disorder because they don't have another X chromosome to basically hide the genetic condition behind. All right. Okay, let's move on to question 3, a pedigree question. So it says the ability to taste PTC comes from a dominant allele. So I'm going to put a big T here for being a PTC taster. And little t, my computer always wants to make it a big T. So not a taster for little t, right? So ability to taste PTC comes from a dominant allele. The shaded in shapes represent individuals who can taste PTC, all right? So every shaded individual here is going to be able to taste PTC. So we want to put a big T within everybody who is shaded in, right? Because they can taste it. So we know they have the big T allele, right? Now, what about our unshaded individuals? The ones that have the clear centers here. All right, so they cannot taste it at all, right? Because the shaded in represents those who can taste. Tasting is controlled by a dominant allele. So if you're not a taster, then you have to have 2 recessive alleles. All right, let's see if I can, oops, I guess I need to change my font here, color. There we go. Okay, so there's only one genotype then if you cannot taste, right? So all of these individuals that are not shaded in will be homozygous recessive for not being a taster. Now, keep in mind, this doesn't say anything about it being X-linked, so you don't have to worry about whether they're male or a female in this case. Now, typically with a Punnett square, the males are the squares and the females are the circles. All right, and I apologize, my line. I shifted everything down here. My line didn't make it all the way down. Okay, so now let's go back and look at our shaded in squares. So we have a dominant allele for this parent here. So this is the mom, this is the dad. The line going down represents all their children then, right? So this line across represents a union between this individual and this individual, and now they have children which are seen in that next generation. So if we follow it down, they've got a son, they've got a daughter, they've got another son, another daughter, and another son. And this individual is just hanging out there because I forgot to draw the line connecting them. But we can make them another daughter if we want. And put them in the family there. Okay? All right. So this son here, shaded in, he's a taster, but we want to know what his other allele is. So he is the product of these 2 parents. This parent is only going to be able to pass on a recessive allele. So therefore, the genotype of that individual has to be, or that son has to be heterozygous. Because this parent here, the father, can only pass on the recessive allele. They got their dominant allele from their mother. Now let's look at mom. Mom we know is a taster, but she has children who are non-tasters. So in order for that child to be a non-taster, mom must be carrying the recessive allele. All right. So I don't think my font is coming up right here.